Welcome, Dr. Li.
Thank you, David.
Firstly, tell us a little bit about your laboratory and the scope of your work.
Okay, so I'm the principal investigator for the section of retina neurobiology. I'm a basic scientist and working on retina, mostly the neurobiology of the retina. There are several focus that we do in the lab. Some of them are more geared towards synapse, how neurons in the retina talk to each other. And some of them are focused on how neurons connected to each other form kind of a circuits to encode visual information, propagate visual information in the retina. And others, we are also interested in the metabolic aspect of retina, and we study some extreme metabolic conditions and how retina adapt to those conditions. We hope that by studying those, we'll be able to find some strategies that retina can cope with the metabolic challenge, which will be very relevant to the health and function of the retina.
So tell us about some of the leading research that you're working on.
On the synapse side, so retina has this very unique synaptic structure that's known for many years, and we know they're very important for visual function. And this is not limited to the retina. Some other sensory, primary sensory neurons also has it, such as hair cell in the inner ear. They usually are has some function in neurons that has to continuous function and release and cycle their neurotransmitter containing vesicles. This is a structure called synaptic ribbon. So we study the structure and function of that particular photoreceptor ribbon synapse. And we used a model system in which this structure changes, and we study the functional change associated with it. Another example of our research more focus on the circuitry, and we study how color information is processed in the retina. In particular, how the blue light information that received by S-cone or short wavelength sensitive cones, and how these information are propagated through retina.
Very good. I'm just going to hold this. Now, does your unit also look at the immunology of retinal cells?
Yes, in our institute we have a specific laboratory dedicated to that research. Myself, we haven't been working on that end very much. So that's Laboratory of Immunology led by Dr. Robert Nosenblatt. It's a very famous clinician and immunologist in the eye disease.
So you've given us quite a large spectrum of work that you're doing. It's very impressive.
Thank you.
What areas do you believe are the most promising from a translational perspective?
Well, I think all the basic science is very important for us to understand how the retina function, and we ultimately wanted to know how visual information is encoded and propagated and passed along through optic nerve to the brain. And some of those might not be immediately has implication to clinical treatment therapy, but on the long run they will be very useful. In particular, on the translational aspect, I think our work on the metabolism and how retina adapt to some metabolic stress condition, they could potentially have some implication in our hope to deal with conditions where the health of retina is challenged because of metabolic stress.
If you look at your current research, what surprising factors have you uncovered? Is there anything that's really surprised you in the last year amongst the research you've been conducting?
The research overall is always filled with surprises, surprise failure, surprise success. For the last couple of years, we have conducted a study looking at, we study the animal that hibernates in the winter time, and that's a very drastic change metabolically. And we has been focused on some of the changes, for instance, the synaptic change and mitochondria change specifically. But then we also started to do a more systematic approach using such as RNA-seq, proteomics studies, looking at the global changes. So what surprised us when we did the RNA-seq study of the awake versus hibernating animal, looking at that RNA profiling of the retina, we were quite surprised to see how much of a change, the landscape of genes profiling change, which is beyond our imagination. We would imagine more post-translational modification, but in fact there's a lot of change in gene profile, and we're still mining that very heavy data and trying to figure out or at least identify some interesting gene to follow up with.
One of the great areas that people are revisiting now is central nervous system plasticity in the area of potential value of stem cells, taking essentially neural cells that have no further regenerative capacity to enable them to A, regenerate, or B, to have a degree of plasticity, so in the setting of injury, the setting of insult. Are you folks looking at that?
Well, there's different levels of plasticity, and some of them not necessarily involve regeneration of neurons, but they're a component of the neurons, such as synapse. They could have a level of plasticity that we can harness to improve the survival of the neurons, to improve the condition. For instance, in our study, when we look at the animal that hibernates in the wintertime, the retina has shown a level of plasticity that we do not normally see in animals that do not hibernate, and including structure change of synapse, and they appear to have very specific functional consequence in that they could maintain a certain level of signaling function that needed for vision, but also reduce the energy consumed and during that period of time. I think so. These type of plasticity also very useful and could be further studied and looked into. In terms of the regeneration, we are keep an eye on that, and we actually, in fact, generate our own stem cell lines, iPS stem cell, from those hibernating animals in a hope to finding the special factors that render them being more resilient to metabolic challenge conditions. But I certainly have been very keen on the fact that we now have a capability to generate neurons and RPEs from ES or iPS cell, and the application of potential for transplantation and all that. There are still a lot of work needs to be done to figure out how they could integrate into the existing retina neurons and systems and be more function. I think that's really the key.
And one of the areas that people are looking at is looking at hibernating cells. And as you know, in macular degeneration, there's probably a window of hibernation. The cells don't undergo apoptosis, they don't undergo irreversible damage for a period of time. So identifying hibernation is obviously a challenge. Do you folks look at that?
Yeah, very interesting point. That's certainly a different concept of hibernation that we normally think of. But I think probably what I gather from your question is that there are some common themes in those cells' behavior. For instance, they could curtail the metabolic rate, and maybe that's the key for, if we could call them, an hibernating status. And for the animal as a whole, when they hibernate, the key feature is indeed a drastic decrease of metabolic rate. And certainly when tissues are, for instance, retina are challenged with lack of nutrition, oxygen, and certainly it might be a good strategy to tune down their metabolism, to put them through a kind of a low metabolic state before anything could be restored or other medical treatment could be available.
What would you say is the best objective measure of changes in metabolic rate in vivo?
Yeah, that's a very good question, and I think that's what people have been looking for, and that certainly could be very important and useful for in a clinical setting for diagnosis and looking for early signs of changes. We do not have a clear answer to that yet. Certainly something we're interested. For instance, on the imaging side, whether some of the mitochondrial functional change could be visualized as some sort of optical signal, and we are also conducting some OCT studies in hibernating animal as well, identify some of the changes in OCT signal, for instance. But these needs to be further investigated to establish the link to true metabolic change.
Right. Are there any biomarkers that have popped up?
Not at this stage, but I think many labs are working on that.
Excellent. Now to change gears to patients, and the following question is really directed at the general public who will be watching this. What are the potential implications of your work in terms of improved therapies or other options down the line?
Well, I think I'll put it in two levels. Some of our very basic research, looking at the synaptic structures and circuitry, I think is very useful foundations for future therapeutic interventions, such as with stem cell, with gene therapy. We introduce new healthy neurons, for instance, into retina, but they all have to sort of go through a redevelopment stage to correctly incorporate into the system to be functional. So who are they supposed to talk to? How do they talk to the target? How do they find target? These are the things that the basic scientists are gaining knowledge for the future use. So that's one level. On the other level, some of our studies on metabolism, energy related, that could be may have closer applications because under many of the disease conditions we now know that energy is a problem, metabolism is an issue that retina has to cope with. And if we, for instance, our study on hibernating animals, finding some of the molecular mechanism or strategies they take to cope with that kind of a challenge, then we can use those strategies in patients in disease condition when they're facing similar metabolic challenge.
I think not many people are familiar with the fact that the eye has a very high metabolic rate. It's one of the highest in the body, if not the highest in the body. Maybe
Exactly right. Exactly right. The brain is 2% of the body weight, use about 20% of energy, and retina is one of the top, if not more so. In particular, if you think about the retina has to function day and night, we probably naively think when we're sleeping, eyes closed, it's in the dark, retina probably is at rest, but it's actually not quite true. And under those conditions, in the dark, photoreceptor actually also functioning quite hardly at their synapse site.
If you look at the broader spectrum of work being presented at ARVO 2015, is there anything that jumps out at you as being particularly noteworthy?
Well, it's still at the quite early stage, and today, the Sunday that just opened, but already there's lots of interesting symposium and talks. I personally attended the Sunday symposium that we, Visual Neuroscience session, organized with a very good collection of speakers talking about how to regenerate neurons, how to reconnect and rewire, and so that to restore vision. And this is a very long-term ambitious goal that come up with by the community, operated by our institute, and called Audacious Goal. I think those speakers really present the state of art of research, tell us what we at now. We are capable of generating type of neurons we need for receptor RPE, and to some extent we were able to starting to put them into, at least in animal model, put into retina, and also acknowledge the big challenge we're going to facing ahead of us. How those newly introduced cells can be properly wired into the system, and how to generate enough of cell for that. These are the questions generated, but certainly people see the hope, and that's, I think, all the people here are working towards this audacious goal.
How are you rerouting neurons?
In terms of research?
Yeah.
I think we still working on very fundamental basic science research. In fact, one of the factors we discussed is that for many of the regeneration work, basic knowledge of the development of biology is almost key and critical to unveil some of the key factors that could change the efficacy of regeneration, for instance, and even the intrinsic capability of regeneration to unlock that. So I think my work, the couple of areas that we're interested in, we're still going to pursue those. And I think all of the knowledge generated would be very useful at a certain point.
Dr. Li, is there anything else you'd like to add?
Well, I think I really appreciate the Macular News and doing this, and to increase the public awareness of the issue and what research is doing, and possibly maybe getting more support from the private foundation as well as government. As a government employee, I certainly not in a position to lobby the government to increase the funding, but and really the basic science and medical research is important for the health industry, for the everyone.
Dr. Li, it's been a privilege and a pleasure. Thank you so much.
Thank you, David.
Thank you.